Hello, everyone. My name is Fernanda Diaz. I'm an assistant professor of analytical and food chemistry in the Department of Food Science and Nutrition at University of Minnesota, as well as a researcher in the Plant Protein Innovation Center. And it is a pleasure to be here today to talk and discuss a little the challenging topic of off-flavor formation in pea proteins, and I hope to share some insights and shed some light on new approaches that can enhance our understanding in this important issue. As we all know, we face an enormous challenge, that is how to feed an increasing world population that is expected to reach 10 billion people by 2050. And to adequately feed this increasing world population, we need to develop a pathway that will lead us to sustainable food production. We need to think about natural resources such as water, energy, land use, as well as processing strategies that will lead to the production of a healthier and higher quality products, as well as the utilization of food waste. In this sense, alternative proteins, they arise as a sustainable source of protein that meets this consumer desire related to environmentally friendliness, animal welfare, and health aspects. As we can see here, alternative protein consumption has increased drastically in the last decade, which plant-based proteins leading this growth, followed by fermentation, as well as most recently cell-based proteins. So at the Plant Protein Innovation Center, we work with several proteins, alternative plant proteins that are gaining traction, including pulse, such as peas, chickpeas, lentils, and beans, oil seeds, we have canola, sunflower, camelina and pennycress, and other plant proteins, including oats, hemp, and leaves such as alfalfa, and so on. A little bit of our center, we have this mission of bringing together interdisciplinary research and industry partners to deliver to the supply chain new nutrition and functional plant proteins. The Plant Protein Innovation Center, or PPIC, is the first of its kind in the nation, and address industry identified challenges and opportunities to develop this wealth of fundamental knowledge that will lead us to a low emission food industry. We have a diverse research priority areas that will include production, that are focused on breeding, sustainability of the supply chain, as well as diversity, crop diversity, processing and formulation. We have areas on extraction technologies, high value and use for those new crops as well, and application that includes flavor. This is the topic of this presentation, functionality and nutrition as well. And those priorities, those research, they are funded by several different agencies, including FFAR, GFI, USDA, the Minnesota Department of Ag, Forever Green, Institute on Environment, and North Harvest Beans. And we all know that partnering with industry is key to actually tackle those main issues in plant protein. So at PPIC, we come together to grow research. We have a variety of research areas that include analytical chemistry, food science, nutrition, experts in various fields that will include protein chemistry, metabolomics, and so on, and a broad industry instrumentation and research capabilities to bring innovation in the plant protein space. So we work to develop the ideal protein. But what would be the ideal protein? When we think about ideal plant proteins, we think about a highly functional protein with good solubility, emulsification, foaming capacity, all tailored for a specific application. We also think about highly digestible proteins with a complete amino acid profile, reduced content of anti-nutritional factors, reduced allergenicity as well. And another main factor is flavor. Consumers, they want a desirable flavor. Flavor is one of the main drivers for consumer acceptability. An ideal protein would be like this bland protein that would serve as this white canvas for flavorists and processors, food processors, to add different flavors and make a ideal product. But we know that it's not exactly how that works. When we think about flavor as well, we need to think about several different aspects, such as olfaction. We are talking about the sense of smell, aroma, and for plant proteins, we're talking about green, grassy, and beany aroma that is perceived in several plant proteins. Gustation, or the sense of taste, and we do perceive bitterness, for example, in a variety of plant proteins as well. And we also have somatosensory that will encompass astringence of taste as well as the hot and cold sensation that will limit the palatability of our product. Therefore, we need a holistic approach to understand this complexity of flavor as one of the main drivers of liking. So plant proteins, they bring a flavor challenge since they commonly carry flavor components, characteristics of the plant source. We know that the consumers are demanding innovative plant proteins, which creates opportunities as well as huge challenge for flavorists. We know that flavor is key for consumer acceptance and that plant proteins are known for unpleasant flavors. We often describe plant protein flavors as beany, grassy, and as well as we describe some bitter taste that usually come from saponins, oxidized fatty acids, or oxidized lipid oxidation products, as we're going to see, and peptides. When we talk about off-flavor and flavor in plant proteins, we need to consider inherent off-flavor of the proteins that comes from the metabolism of the plant itself, metabolism of lipids, proteins, and carbohydrates. We need to think about those off flavors that are being formed during process, harvesting, storage, as well as plant the protein and flavor interactions that can hinder some flavoring agents that we add to the protein, for example, or can lead to different outcomes. So therefore, we work to develop this comprehensive platform that will assess the effects of breeding, processing, storage on the flavor chemistry and sensory attributes of those alternative protein ingredients. All flavors in plant proteins, they are volatile compounds and they are hydrophobic, which means that during the plant protein extractions, they are not removed, they are not extracted by the water that we use in the process. So they are concentrated in our plant protein. So how do we reduce this off flavor? Another question that arises, how processing affects those compounds? So we need to develop this fundamental understanding of this flavor protein interactions, as well as how fatty acid composition affect our flavor profile to develop better processing strategies to deliver a high quality protein. When we look at the literature relating to aroma compounds and flavors and off flavor found in plant proteins, what we notice is that the majority of those compounds, from 60 to almost 90% of them, they arise from lipid oxidation. They are lipid oxidation products such as aldehydes and alcohols and ketones. We also see some amino acid degradation products as well as Maillard reaction products that will depend more on the type of heat treatment that that protein went through. So we have our isolates. We need to keep in mind that protein isolates are mainly protein, so the main compound is protein, but we do have residual lipids. And those residual lipids, since they are from plant source, they are rich in polyunsaturated fatty acids, such as linoleic acid and alpha-linolenic acid. And those polyunsaturated fatty acids, they are highly susceptible to oxidation. And they will form hydroperoxides, and later on, they will form secondary products of oxidation or volatile compounds that are those responsible for the beany, greeny, grassy aroma that we perceive. And just to list off a few of those compounds, hexenal, heptanal, nonenal like aldehydes, some alcohols, some ketones, and so on. Just to remember, lipid oxidation is a very, very complex reaction and is associated with the generation of off-flavors that will change the sensory attributes, will reduce the shelf life of our products. And those oxidized lipids that are also a health concern, and they reduce the bioavailability of some essential fatty acids as well. We have what we call primary oxidation products that are usually non-volatile products that are generated during this complex process. And here, just to illustrate, our linoleic acid that plant proteins are rich in generates some hydroperoxides, some hydroxides, and some ketone forms. They're all main contributors to later on formation of off-flavor. And as we see here, our linoleic acids form our primary products, also collectively known as oxylipins. That's how I'm going to refer to them. And our volatile products here, some aldehydes, such as the 2,4-decadeenal, octanal, and hexanal, that will give that green, grassy, sharpy, waxy, leafy odor to our plant protein. And when we think about lipid oxidation, we need to keep in mind that it's hard to detect those compounds with standard methods. It's hard to predict and therefore stop oxidation during processing. And therefore, this will imply in problems with stability and shelf life. Our goal is to help understanding when, where, and how those products arrive, what are their chemical composition, and how they affect off-flavor formation in plant proteins. So our lab focuses on a holistic approach where we look at esterified and free fatty acid composition, primary oxidation products, and secondary oxidation products, and their relationship using analytical techniques such as gas chromatography and mass spectrometry, or liquid chromatography with mass spectrometry in tandem, to understand how processing harvest will affect the formation of those off-flavor compounds. And I brought you one case study of one project that we have been developing just to illustrate how we can apply this concept in to enhance our understanding in the off-flavor formation in pea protein. Our goal here was to understand how fatty acid composition would drive the formation of oxylipins that are primary oxidation products and volatile compounds during pea protein extraction and isolation process. We used 6 samples, and 2 winter peas, 1 flour, and 1 pea protein isolate. 2 commercial samples is the flour and the protein isolate, as well as the spring pea, flour, and isolate. Here is the spring pea and the winter pea that were cultivated here at the University of Minnesota by our Forever Green initiative, and the commercial samples were donated by industry. We therefore analyzed fatty acid composition, primary products, namely oxylipins, secondary products of flavor compounds or volatile profile, as well as statistical analysis to better understand our or interpreted the data, understanding the data, as well as the fat, total fat content. Related to the fatty acid profile, what we observed is that, as expected, all the samples the main compound was basically our linoleic acid with an omega-6 essential fatty acid, followed by our oleic acid with monounsaturated fatty acid, as well as our linolenic acid, alpha linolenic acid, and omega-3, which means that those samples, their residual lipid is highly prone to oxidation as expected. And here, just to illustrate the lipid content, our flour ranged from 2%, our commercial and winter flour, where our spring flour was slightly higher in lipid. And the plant protein process actually increased our lipid content. We need to keep in mind that peas are usually not defatted, or mainly not defatted, while processing for pea protein isolate. Looking at fatty acid composition now, quantification in milligrams of fatty acid per gram of sample. We see here our main fatty acids, palmitic, oleic, stearic, oleic, linoleic, and alpha-linolenic. And if we zoom here in the two ones that are more susceptible to lipid oxidation, we see that the plant protein samples, they have higher concentration of those compounds as expected. And we do see that our commercial had a higher concentration, followed by winter and spring pea protein isolate. Before we start our data on oxylipins, I just would like to clarify some concepts. When we talk about 3-acetylglycerol, we talk about a glycerol backbone bound via ester bond to 3 fatty acids. This 3-acetylglycerol structure can be hydrolyzed to release free fatty acids. This 3-acetylglycerol can be oxidized to generate what we call esterified oxidation products or oxylipins that can also be broken down to generate what we call free oxylipins. The same pathway can go from free fatty acids to free oxylipins, which means that they can be oxidized, and both of them will lead to the formation of off-flavor compounds or secondary oxidation products. Here to illustrate, all our samples, they have a majority of their oxidation products in the esterified form, which means that they were attached to the 3-acetylglycerol backbone. And we have some free fatty acids here. What is the relevance of this? The relevance is that we usually look at free fatty acids as the main source of oxidation, which is true. But given the amount of esterified oxidative products that we have, we should also take a look in this pool and try to understand how this pool would affect the formation of off-flavor. Looking at here, this is our total esterified plus free oxylipin profile. Here, in a simplified manner, we see that spring pea had the highest component, highest concentration, followed by our commercial and winter pea protein isolate, with the flowers again with a lower concentration. This increase, of course, is due to the higher lipid content, but as well as is highly affected by process as well. The majority of the esterified oxylipins that we found were derived from the linoleic acid, that is the main polyunsaturated fatty acid. We have some complex names here, but just to illustrate, some hydroxy forms were the main compounds, some ketone forms that are actually derived from this compound, and triol forms were found, and there is some studies trying to correlate this with bitterness as well. We see some free oxylipins, similar pattern here. The main difference is our commercial sample is not as high. Here could be for two reasons, that it is not as oxidized, but also that it already oxidized to a volatile compound. And that's what we're gonna see looking at the volatile profile. And we also notice that the majority of those compounds, they are derived from the linoleic acid. We see ketone forms here, which means that our hydroxy forms probably were already degraded. So the oxidation was happening probably faster in this free pool, as well as some triols. Looking now at the volatile compounds, we saw the alcohols, aldehydes, and ketones. Like again, lipid oxidation products, they constitute more than 60%. That is in accordance with what we see in literature for plant proteins. So 60% of all the volatile profiling. And here, just to highlight some of the main ones, we see here our hexanal that brings that greeny, grassy aroma to our plant proteins, spring peas, a higher concentration found, followed by commercial, and winter pea protein. Again, all of our flowers are a significant lower concentration. We see highest amount of benzaldehyde that brings almond and cherry-like notes to our protein in our commercial pea protein isolate. Looking at alcohols, our main alcohol, that is 1-octane triol, that brings that hay-like and earthy-like aroma, very high in the commercial sample, which correlates with a low amount of free oxylipins found in the samples, as well as some ketones that are responsible for green aroma. We also see higher concentration in our commercial, and here higher concentration in our spring pea, as well as commercial plant protein isolate. We did also principal component analysis where we noticed that PC1 plus PC2 was able to explain the difference of our samples in about 60% of this difference. We kind of see a cluster here of our plant protein isolate and a cluster of our flowers. Of course, here we have a very high proximity of our winter pea flower and commercial pea protein isolate. When we look here at our heat map, we can see that our spring pea protein has high concentration of volatile compounds as well as oxylipins. Our winter pea has more of this oxylipins profile. Our flower, winter pea protein, winter pea flower is very clean, which is very interesting. So we see the impact here of processing with the increase of those oxidized compounds. Our commercial pea protein is rich in some linolenic acid derived oxylipins here as well. The flower has some of the oxidized products, and our spring flower has well some oxidized products, but all very different profile. Just to conclude this brief work, that is the first work being developed in this area, so it's everything very new. We are still correlating how those compounds are formed, trying to find exactly all the pathways that will lead us to a better understanding and of the process, and can help us also to guide new processing that will deliver a protein with reduced oxide information. So what we conclude is that protein extraction and isolation significantly increase the total oxylipins and volatile formation in pea protein isolates, leading to this notable rise in off-flavor markers. So now we need to explore strategies to reduce this residual lipid in those protein isolates or improve process conditions that will minimize the lipid oxidation to enhance the consumer acceptability and quality of those plant proteins. We also noticed that this oxylipin profile were positively correlated with off-flavor formation. And why this is important? Because this approach that is based on a targeted lipidomic approach to quantify oxylipins, since it's so sensitive and it comes before off-flavor, it can be used to review new early lipid oxidation markers, as well as fingerprints of processing and their impact on off-flavor formation. With this work, we also conclude that the off-flavor issue in plant protein requires a collaborative multi-phase research to be solved, because it's a very complex issue. It's not a one-man job. And we also know that with this variety of new and increasingly popularity of plant proteins, we have several opportunities, new challenges that will drive innovation in this space, but flavor will continue to be essential to the success of the new products being launched, as well as to remain competitive in this marketplace. And I would also like to acknowledge Dr. Pen Ismail, that is the director of the Plant Protein Innovation Center and a partner in this project that is being funded by the Forever Green Initiative, and my postdoc, Dr. Wellington Oliveira, as well as my lab members, and the University of Minnesota for all the support. And I would like to end this presentation telling you to partner with us today to change the landscape of plant protein tomorrow. Here are more information on our center and our website, as well as my email, in case you have any questions. Thank you.